Okay, hello everyone. Welcome back. This is video 3 in our unit 1 of biology leap remediation. And today we're going to talk about protein synthesis. And anytime you see the word synthesis, that means that you are putting things together to make something. So like a synthesis essay would be to synthesize the essay would be to put all the pieces of your essay together. So in this sense, we're talking about putting amino acids together to make a protein. So that is protein synthesis. And DNA and RNA are the key players in this process. And so just as a quick review, remember in our last video, we learned about DNA and RNA. We learned what they look like, what their individual components are and how they're different. And then prior to that, we learned that DNA and RNA are examples of nucleic acids, which is one of the 4 major macromolecules found in all living things. So remember we talked about that living things are made up of the molecules of life. And there are 4 major categories of these molecules of life, also called macromolecules. And there's proteins, carbohydrates, lipids, and nucleic acids. And we get our carbohydrates and our lipids primarily, and proteins from food, but the nucleic acids have, we get the components for those from food as well, but the nucleic acids have a very different job. And that's what we're going to learn about today. So our nucleic acids are DNA and RNA, and they're made up of these nitrogenous bases, also called nucleobases. Cytosine, guanine, adenine, and thymine in DNA. And of course, there's no thymine in RNA. Instead, RNA has uracil. And so we learned that DNA is where the genetic information is stored. It's in the nucleus of the cell. And then we learned that RNA controls the expression of the genetic information, and it goes out of the nucleus into the cytoplasm. And so we are going to talk more in detail about that process where DNA makes RNA and RNA goes out into the cell and makes a protein. And so protein synthesis is considered the central dogma of biology. Francis Crick was one of the scientists that discovered the structure of DNA, this double helix spiral staircase looking structure. And from discovering that structure, that scientists were able to explain how the information that is held in the DNA, the genetic information, flows from that DNA to other parts of the cell, and then ultimately to all parts of the living organism. And so we learned that all of your cells have a complete copy of your DNA. And we know that cells can make a copy of themselves. And that's how we make new cells in our body. And when our cells are going to make a copy of themselves, they need to make a copy of their DNA as well, so that when they split in half and become 2 cells, each of the 2 cells will have a complete copy of the DNA. And now the process of DNA making a copy of itself is called replication. Okay, and so when DNA replicates, it unzips, kind of like a zipper. The 2 strands come apart, and when they come apart, the replication takes place, And you remember yesterday we talked about the base pairing rule, the complementary base pairing rule. So when DNA replicates itself, there is a base pairing rule that is in effect. And so anywhere on the original DNA strand where there is an A, which stands for adenine, then it is going to be paired with a T that stands for thymine. So A goes with T, and then T goes with A every time. And so you can remember apple in the tree. And then there's two other nitrogenous bases. There's cytosine and guanine, and they go together. Cytosine on one side gives you a guanine on the other, and vice versa. So remember car in the garage. So that is for DNA replication. So when DNA is making a copy of itself. And then the processes that we're really going to dive into today is the process of protein synthesis. Protein synthesis has two steps. The first step is called transcription. Transcription is when DNA makes RNA. And remember, RNA, instead of being a double-stranded spiral helix, it is just a single strand. And then we learned that RNA leaves the nucleus because DNA cannot. DNA has the genetic information, the genetic code, but it cannot leave the nucleus, so it makes RNA instead. RNA leaves the nucleus and it goes out into the cytoplasm to the ribosomes. And if you'll recall, ribosomes are little organelles inside of the cells, and they make proteins. So the RNA takes the code for how to make a protein out of the nucleus, finds the ribosome, and from there, the second step of protein synthesis takes place, and that is called translation. Translation is when the RNA code is translated by the ribosome into a protein. And remember, proteins are made up of amino acids. So when you see this little chain of blue circles here, each of the little blue circles is an amino acid. And if you'll remember back here where we talked about the macromolecules or the organic compounds, we learned that proteins are long chains of amino acids. So the monomer, or the single unit for a protein, is an amino acid. And so proteins are made up of many amino acids linked together. And so that is the process that is called translation, when the amino acids are linked together to form the protein. And that happens at the ribosome. So this whole process can be summed up just in a few words. So protein synthesis is when DNA undergoes transcription to produce RNA. And there's a specific type of RNA that is produced here in this process. It's called messenger RNA. So we just put the little M in front of the messenger RNA, and that tells us the type of RNA. And we'll get into the different types of RNA later, but just know that that particular type, the one that is made in transcription, is called messenger RNA. And then the RNA undergoes translation to produce a protein. Okay, so that's the whole process summed up in just a few words. DNA undergoes transcription to produce messenger RNA. RNA undergoes translation to produce a protein. That is protein synthesis. Okay, so let's look at it a little more in depth. So first, we're going to list and describe the two steps of protein synthesis, and they both start with a T. So step one is transcription. And in transcription, in the nucleus, DNA makes messenger RNA. That's it. That's all transcription is. And then in step 2, which is called translation, the messenger RNA goes out of the nucleus into the cytoplasm to a ribosome, which reads the code and builds the protein by connecting amino acids together. Okay. Step 1, transcription. It happens in the nucleus. DNA makes RNA. Step 2, translation. The messenger RNA goes out of the nucleus into the cytoplasm. It goes to the ribosome. There, the ribosome reads the code that is on the messenger RNA, and it builds the protein by connecting amino acids together. So what is the purpose of protein synthesis? It is to build proteins according to the code in the DNA. Okay, so proteins have to be made in a very specific pattern. Certain amino acids have to go together in the right order, and then the protein will fold into a distinctive shape. And if anything goes wrong in that process, then the protein will not work correctly. And so it's very important that the code is read correctly and the protein is built the way it's supposed to be built. Okay, so now let's do a little practice. Now, the first thing I want us to practice, because I want you to be able to distinguish the difference between DNA making a copy of itself or DNA replication, and then DNA making messenger RNA, which is transcription. Okay, so first we're going to practice DNA replication. And so remember that when DNA is replicating itself, it is making another strand of DNA. So it's DNA to DNA. So the base pairing rule is that adenine goes with thymine, cytosine goes with guanine. Remember, apple in the tree, car in the garage. Also remember that we use an A, a capital A for adenine, capital T for thymine, capital C for cytosine, capital G for guanine. Okay, so when DNA is making a copy of itself, A goes with T, T goes with A goes with T. So ATA becomes TAT. TAG becomes ATC. ATT becomes TAA. ACA becomes TGT. Okay, I've got one more here for you to practice. So if you will pause the video and do number 2 on your own, and then come back and we will check your answer. Okay, so let's check your answer. So TAT will be ATA. TAG will be ATC. ATT will be TAA. ACA will be TGT. Did you get it right? Okay, so let's go on to protein synthesis now. And remember that protein synthesis is making the RNA. So, or the first step in protein synthesis is making the RNA, which is called transcription. So, for number 3, we are going to, for number 3, we're going to transcribe the DNA strand that you see here. And now remember, whenever DNA is making RNA, we have a different base pairing rule. And so you'll see, if you'll remember back here in our chart, when DNA is going to RNA, this is called transcription, and we have a little bit different code. And you will need to remember this code. You need to commit it to memory, because on the LEAP, they won't give you this information. They expect you to know it. Okay, so A goes with U instead of with T, because remember, let's look at this. Remember, RNA, come on, work with me here. So, goodness, sorry. Okay, so if you'll remember, RNA, try it again, does not have thymine. Only DNA has thymine. And so RNA has uracil instead. Let me make this just a little bit smaller so maybe you can see it. Yeah. Okay. So you see how DNA and RNA have cytosine, guanine, and adenine. They have those in common, but DNA has thymine and RNA has uracil. Okay, so let's go back down here and let's do a little practicing on the first step of protein synthesis, transcription. So if our DNA strand is, our first 3 letters is TAT, we know that T goes with A and A goes with U. So TAT will be AUA. Okay, now TAG, T goes with A goes with U, G goes with C. So TAG becomes AUC. ATT will be UAA, and ACA will be UGU. Okay, so that's how you do it. So I want you to pause your video right now and do this one for practice, this one that is down here. So you're going to take this DNA and do the same thing we did up here and change it over to messenger RNA. And here's your code right here. But as you're doing this, see if you can do it from memory, okay? Because you have to have it memorized. Okay, pause the video and practice. Okay, so let's see if you got it right. AAG is going to be UUC. CCT is going to be GGA. TCG will be AGC. And ATC will be UAG. Okay, is that right? Get it right? Okay. So every 3 letters of the DNA to the RNA is called a codon, and that simply means it's a code. Okay, so that's why we've been writing it like this with 3 letters together and spaces in between them, not only to help us to not get confused with all these letters running on together, but each 3 letters has a significant code that we need to be able to work with. Okay, so it's called a codon. And it is used in the second step of protein synthesis, which is called translation. And in translation, we use this amino acid chart. Now, there are two types of charts. There's the square chart like this, and then there's also a circle chart that you've probably seen before. But we're going to use the square chart today in our practice. Okay, so since each of these three letters is a codon, it is a code for a specific amino acid. So when you look in this chart, you'll see you've got your codons over here, the AUU, AUC, and all those are your codons. And then over here on the right, beside the codons, are the names of the amino acids or the abbreviations. So like LEU is the abbreviation for the amino acid leucine, and PRO is the abbreviation for the amino acid proline. So each of these little abbreviations is an amino acid, okay? So let's do a little bit of practice with translation. So the first codon we're going to translate is UUA. So we come down here to our chart, and we see that we find our first letter on the left side of the chart. We find our second letter across the top of the chart, and then we find our third letter on the right side of the chart. So we're looking for UUA. So my first letter is U. So it's going to be somewhere in this row. Okay. Now my second letter is also U. So here's U across the top would be this column here. But we're looking at where do they intersect. So they intersect right here in this first box. So UUA is right here. We don't even have to come over here to the third letter, but we can. We can find A right there and scroll over this way. And UUA is leucine. And we don't have to remember the words for the amino acids. They're big science words. We just are going to type the abbreviation, which is LEU. Okay. Now, the next amino acid that we're going to decode by using the codon is CAG. So let's look at our chart, and our first letter is C, and then our second letter is A, CA. So they intersect right here in this box. So my third letter is G, CAG is GIN. The abbreviation for the amino acid is GIN, GIN. Okay. So now I am going to give you an amino acid sequence, and a messenger RNA. Not an amino acid sequence. I'm going to give you a messenger RNA sequence, and then from there, you are going to get, I want you to practice getting the amino acids. Okay, so we'll have CC, U, G, U. Okay, so now you pause the video and you figure out what the amino acids are for these 4 codons. Okay. Okay, you ready? All right, so CCU is our first one. So our first letter is C. Our second letter is C. So they intersect here in this box. So CCU is proline. So we're going to abbreviate just PRO. And then UGU, U is my first letter, G is my second letter, and then UGU is cysteine, CYS. AGA, my first letter is A, my second letter is G, so they intersect right here. AGA, here's my A, AGA is arginine, ARG. And then UAA, first letter is U, second letter is A, so they intersect in this box. And UAA is going to be stop. So the stop codon is important because it tells the ribosome that it is finished. So when the ribosome gets to a stop codon, then it knows that that protein is finished. So it releases the protein from the ribosome, and then the protein goes off into the cytoplasm. It folds up in a very distinctive way, and it goes off and it does whatever job it's supposed to do in the body. Okay, so let's do a little bit more practice now. So I want you to pause the video, and I want you to do number 7. Number 7 has 2 parts. First, you're going to do transcription and change DNA to RNA. And then you're going to do translation and use the chart up here to figure out what the amino acid sequence is for the protein. And then I want you to keep going. Before you come back, I want you to go ahead and try this LEAP question. It's the same exact skills as what we were just going over, but the LEAP style questions look a little different. And if you've never seen one before, you might get a little confused. So I want you to try the LEAP style question. And there are 3 parts to that. Okay, so go ahead and pause the video and then come back and we'll go over your answers. Okay, here we go. Let's see if you got it right. Excuse me. Okay, so first we're going to change the DNA to RNA. This is called transcription. So remember our code. If you forget the code, it's up here, right here. A goes with U, T with A, C with G, G with C. Remember, you have to memorize that. So TAC is going to become AUG. GTA is going to become CAU. ACG is going to become UGC. CTG is going to become GAC. And ATT is going to become UAA. Okay. One way to check yourself when you're doing this is when you finish your RNA, look at it and make sure you don't have any T's, because that's the most common error people make. No T's, no thymine in RNA. So look back over it, make sure you don't have any T's, and then you'll know that you probably got it right. All right, now let's figure out what the amino acid sequence is for these RNA codons. So AUG, so AUG is methionine, MET. CAU, so CAU is histine. HIS. UGC, so UGC is cysteine. GAC. So GAC is asparagine or ASP. And then UAA is stop. Okay. Now let's move on and try this LEAP style question. One of the things that you will find with the LEAP questions is often they give you passages to read that go along with your questions. And you will find that sometimes the information in the passage is not needed in order to answer the question. But you still need to read the passage because sometimes there is information. Some of the information you need and some of it you don't need. And so they're testing your ability to read and sort through information and figure out what you need and what you don't need. Kind of like when you are doing a Google search and you type in a question and lots of answers come up sometimes, and you have to figure out which answer is the correct answer or which one is the best answer. It's kind of like that. You're developing, you're showing your skill in that area. Okay, so let's read the passage. Jamie inherited a sickle cell trait from her mom and her dad, which resulted in her having sickle cell anemia. She requires blood transfusions regularly to keep her cells supplied with the oxygen they need to maintain homeostasis. The sequence below shows a part of that genetic code for the HBB gene, which provides the instructions for making beta globin protein that efficiently binds to oxygen. So you've got this one particular protein that binds to oxygen, and it's found in your blood. And the information for how to make that protein is on a particular gene called the HBB gene. Okay, and so when you look down here, you see that we have DNA of HbA, which is hemoglobin in normal adults. And then we have down here DNA of HbS, which is hemoglobin in individuals with sickle cell anemia. And so if you look at the two DNA strands, you can tell pretty quickly what the difference is. So you see that the nitrogenous bases are the same everywhere. TGA, TGA, GGA, GGA, until we get right here to the second to the last codon. And here we see we got CTT in the normal hemoglobin, but down here in the sickle cell, we've got CAT. And so that is what's causing our problem. And so from here, we can answer our questions. And we know that we're going to be using our amino acid chart, or they wouldn't have provided it. And so let's go down here and look at part A question. So now this stuff up here in this box, I put that there so that it would be ready for you, because you are going to have to do that. But in the LEAP, they won't give you that. You'll have to know that I need to do that. Okay. So in the LEAP, they'll just give you a question like this. So let's look at what the question is asking, and then you'll see why you need this. So which of the following best represents the amino acid sequence found in normal adults with the HBB gene? And so we've got amino acids here in our answer choices. Okay, but then up here, we've got nitrogenous bases in sets of 3. Okay, so we have got to figure out what the amino acids are. Now, be careful on this, because your first instinct, so we're talking about the normal adults with the HBB gene, And so we go up here to hemoglobin in normal adults, and we're tempted to go, okay, CAC is my first codon. So I'm going to come over here and I'm going to say C is my first letter, A is my second letter. So CAC is right here. And so histidine is my first amino acid. Well, if I look at my choices, I see choice A has histidine first. And if you're not careful, that's what you're going to pick, and you're going to get it wrong. Because if you'll look up here at this DNA sequence, you'll see there are T's. There's a T there, one there, two there, one there. And you can't use T's with this chart. See, there's no T's, absolutely no T's. And that's because before you can use this chart, you have to transcribe the DNA over to RNA. So that is why you have this down here. Okay, but keep in mind on the LEAP, they're not going to remind you of that. You're going to have to remember that. Okay, so the first thing we need to do is we need to take this normal DNA strand, normal hemoglobin DNA strand, and change it, transcribe it to RNA. So CAC is going to be GUG. GTG is going to be CAC. GAC, I'm right here. GAC is going to be CUG. TGA is going to be ACU. Yeah, I'm right here now. GGA is going to be CCU. CTT will be GAA. And then the last one right here, CTC will be GAG. Okay. Now I can use my amino acid chart and figure out what the amino acid sequence is. Okay. And so let's look at our question. We want to know the correct amino acid sequence in normal adults. Okay, and we know it's not A. We've eliminated A. Okay, so let's go ahead and start our amino acid sequence. So GUG, so here's G, my first letter, U, my second letter, and right here in this box is where they intersect. So GUG is valine. VAL. Okay, now if I look at my choices, I know it's either B or C. Okay, so I can eliminate D. Already eliminated A. All right, let's go on to the next codon, CAC. So my first letter is C, second letter is A. CAC right here is histine, HIS. And now I have my answer. I don't have to go through all of these because now with my second amino acid, I know that the correct response is B. So I can go ahead and make that my answer. I'm going to highlight it yellow. Okay. So sometimes, especially if you're in a hurry, you can not do the whole thing and figure out your answer. But you'll see on this next part, part B, we're going to have to do the whole thing. Okay, so let's go ahead and go to B and see what the question says. It says, which of the following can you conclude about the hemoglobin in individuals with sickle cell anemia? Choose two answers that apply. So it says the amino acid sequence for the abnormal hemoglobin replaced a glucine for a leucine. Okay, so we need to know what the amino acid sequence is for the sickle cell anemia. So I've got to go back up here, and remember, I cannot use my DNA. So this is my DNA for sickle cell. I have to change my DNA. I have to transcribe my DNA to RNA first. So the good thing is I already have recognized that these are all the same until I get to right here. So I can just copy this, except for this second to the last one, which is going to be wrong. So my CAC becomes GUG, GTG becomes CAC, GAC becomes CUG, TGA becomes ACU, GGA becomes CCU. And now here's the one that's different right here. So CAT is going to be GUA. And then my last one will be the same. CTC will be GAG. Okay. And now I already have my first two amino acids, valine, because GUG is the same here. And then CAC is also the same. So my second amino acid is histidine. Okay. So, but now I got to do the rest of them because I've got to figure out what it's saying here. It's what was replaced with what. Okay. So CUG, if we go up here and look at my chart, my first letter is C, second letter is U. So CUG is leucine. So I know both of these now are leucine. So I can put it in both places. And then ACU, here's A, here's C. So ACU is threonine. So I can put that in both places because they are the same. CCU, so C is my first letter, C is my second letter, CCU is proline. And I can put that in both places. Okay, and then I'm going to go ahead and skip over here and do GAG because it is the same in both. So G, and my second letter is A. So GAG is glutamine. So GLU here, and then also GLU here. Okay, so this is where they're different. So let me go ahead and figure out what the normal amino acid would be. So GAA, so first letter is G, second letter is A. So they intersect here. GAA is glutamine. Okay, but then GUA, first letter G, second letter U, right here they intersect. GUA is valine. So there's our difference. All right, so now let's look at our answer choices. All right, so answer choice A says the amino acid sequence for the abnormal hemoglobin replaced glycine for leucine. So if we look at the abnormal, the leucine was the same. Leucine is not anywhere else. So leucine in the normal, leucine in the abnormal, so it's not A. Let's look at B. The amino acid sequence for the abnormal hemoglobin was the same as it was for the normal. Well, we know that's not true because we saw right here there was a difference in the amino acids. There was an error in transcription from DNA to RNA. Well, it looks there was an error. There was definitely an error, and it happened right here, right here. Okay, so let's read the rest of our choices because it looks like that might be the right choice, but let's read the rest of them. D says the amino acid sequence for the abnormal hemoglobin replaced a glucine for a valine. All right, so let's look at what we did here. Oh, that's what happened. The abnormal replaced what should have been a glucine with a valine. Okay. And then, so I know that one's right. I'm going to go ahead and highlight that one. That one is definitely right. But I know that I have to choose two correct answers. And so I'm kind of thinking C could be correct. But let's read E. E says there was an error in the nucleotide sequence in the original DNA strand. Okay, so where did the error occur? Did it occur when we transcribed DNA to RNA, or was it already there? And so if we look up here, I can see that the DNA was supposed to be CTT right here in the sequence, but instead it was CAT. So the error had already occurred in the DNA before we ever transcribed and translated into the protein. So the error occurred way back here in the DNA. So that means E is your other correct response. So the correct answers are D and E. And then finally, the last question, you probably wouldn't have known this one unless you remembered, because this is, again, is a review. But in the next video, we're going to talk about mutations. And so we will talk about the type of mutation that occurred here. So part C says, what type of mutation occurred in the DNA of people with sickle cell anemia? If you look up here at the DNA, and you see that the only difference is that instead of CTT right here in the sequence, we've got CAT. And so only one nitrogenous base was wrong, and it was called a substitution because instead of a T, an A was substituted. So that is called a substitution mutation, and you'll learn more about that in video 4. Thank you for your attention.